We want to now start a new series which is about rashes, and the motivation for this is that rashes just pop up very frequently when you study microbiology, when you study viruses, but also when you study bacteria. I'm not going to give examples now because this is then going to be a task for you in our priming to think about cases where you have encountered this, that you see rashes as symptoms for certain viral or bacterial infections. And we'll get going with part 1 now, which is on Meningococcemia and Purpura, more specifically Purpura fulminans. Before we start, it's the early days of 2026 now, and towards the end of 2025, I was really more and more thinking, okay, is this actually still timely to do this kind of, video lectures? Aren't there much more advanced ways of studying nowadays? So one thing that I will talk to you a little bit at the end of this video is an attempt at making a learning app or a teaching app with Google AI Studio. The idea then being, okay, you can make teaching much more personalized than watching videos. And overall, I have to say, well, the technology is amazing. I mean, it has come definitely a long way. On the other hand side, I think we're not quite there yet to work efficiently in this way. But I would be very interested what your comments are, or maybe you have suggestions how to make this better and more efficiently. And for sure, the future will go more and more in this direction of personalized learning. But as I said, let's talk a little bit about this at the end of the video. Okay, so here is a bit for you to think about before we start. So the first one is the one that I just alluded to. Which bacteria, maybe if you know viruses as well, which viruses cause rashes, and describe the rash. So I'm assuming if you have studied microbiology, some things will come to mind. If not, then that's okay, then we'll go through that in the following. And if you can, also think about what could be the underlying mechanism for these rashes. And this is something that I find usually is not explained, at least not explained very much in the textbooks and in the lectures. So think about it and don't feel bad if you can't come up with anything really. Hit pause and think about these questions. Yeah, so to answer about which rashes do you know, it obviously would be helpful to first define what is a rash. If you want, I'll give you a little bit of time so that you can again pause here and think about it yourself. If not, let's go for the definition. And the definition, just taken from Wikipedia, is a rash is a change of the skin that affects its color, appearance, or texture. So very broad definition, essentially pretty much every change in the skin that you could imagine. But among others, what Wikipedia also says, the rash may be localized in one part of the body or affect all the skin. So I won't necessarily make this part of the definition, but something that's good to keep in mind. So here we have a couple of examples for rashes, and admittedly, I did bias this towards rashes with an infection as an underlying cause. So we have, for example, cellulitis, which is bacteria caused, mainly purpura, which is bacteria caused in many cases, measles, which is of course viral. Then here contact dermatitis, this is now not with an underlying infection, but a hypersensitivity reaction to something that comes in contact with the skin. Syphilis, bacterial again. Tinea, so fungal. Fifth disease is again viral. Rocky Mountain spotted fever, this is bacterial again. Pityriasis, which is fungal, and impetigo, which is again bacterial. So as I said, biased towards infectious rashes, but the idea here is just that you get an idea of how varied rashes can be. In many cases, there is a reddening as a change of color. Here in the case of pityriasis, it's actually a loss of color. Then in some cases there are elevations here as well for syphilis. Yeah, so in general, just to give you an idea what different forms rashes may take. Okay, let's answer the first priming question together, and then for the second one, for the causes underlying the rashes, this is then obviously the topic of this and of future lectures, to really think what's the mechanism behind those rashes. But for now, we'll just talk about which bacteria cause rashes. We're not going to go into the viruses. This would lead us a bit far, and we'll look at some picture that kind of describes what they look like. So let's get started with rashes typically associated with Staph aureus and Strep pyogenes. So we see cellulitis here, and we see impetigo here, impetigo contagiosa in this case. Strep pyogenes is a very important one in terms of rashes. So in addition to these ones, there's also this so-called sandpaper rash or sandpaper-like rash associated with scarlet fever, which is a bit hard to see, but essentially it has this sandpaper-like appearance. Then there are erysipelas. Erysipelas are typically differentiated from cellulitis by saying they are more well demarcated, so there's a clearer distinction between the rash itself and the healthy, unaffected skin. Then what we already saw before also was this Rocky Mountain spotted fever appearance, so caused by rickettsia, for example. Or we might think about what is then going to be the topic of this lecture, Purpura fulminans, caused by Neisseria meningitidis in most cases, and Neisseria meningitidis should be the first thing that you think when you see this Purpura fulminans. You might think about some other ones, so you might think about Borrelia, for example. So there we have this this bullseye appearance, which you can also call a rash by our definition, and typical for Lyme disease, for Borrelia. What we have seen before was this Treponema pallidum, so this syphilis, more precisely symptom associated with secondary syphilis, where you have this, I would describe them as vesicles, especially on the extremities, especially on the palms of the hand. Or you might think about Salmonella typhi, you maybe have heard. It's often referred to as a petechial rash or as roseole. And if you have studied a lot of microbiology and think a bit further, you might even come across other ones. So this is not an exhaustive list. And so now the questions that we want to ask: how do the bacteria cause these different rashes? Why is the one rash different from another one in its appearance? Before we now try to go into one specific rash, though, and try to explain the causes behind it, I think it's still useful if we do a bit of terminology. This is not meant as making our lives harder and having to remember a lot of new stuff, but these are terms that you will come across, or that we will also use when we talk more about rashes. So I think it is helpful to look at some of these terms. There are many different ways in which you can have such a decision tree, such a classification to end up in the different terms. I find this one here very useful, but as I said, there are other ones as well where you make other kinds of decisions. And I find this first decision especially useful to divide rashes into blanching or non-blanching, or I guess the proper French way of saying it would be blanching, but we'll keep with how it's said generally in the English-speaking world. And what this one means is blanching means the redness goes away when you press on this red. So you have an erythematous rash, so a red rash, and then you press on it and the redness will disappear, whereas this is not the case in the non-blanching ones, so there the redness does not go away. The subdivision here among the non-blanching ones, this is essentially based on size. We'll see that later on again. And so the medium size, what most, so sometimes this is also incorrectly referred to, or people don't take so much care about the size, then they would generally talk about the purpura. But if you want to be very exact, if they are only very small spots, or small red spots, then you would talk rather about petechiae. And if they're big, then you would talk about the ecchymosis. Okay, let's go to the blanching tree. Again, today we will stay here in the non-blanching side, and you will also see why this distinction is important between if the redness goes away or not when you press. But so for the blanching ones, we have subdivision in, are they either just flat? So in this case, you would have the redness, but not the elevation of the skin. Or is there elevation of the skin, but essentially it's just a solid elevation, so there is nothing, no pus or liquid beneath it. And if it's just flat, then again, this is a decision based on the size. You would either talk about a macule or a patch. If it's solid, then you would talk about a papule, or if it's larger, you may talk about a plaque. If it's pus-filled, it's called pustule. So this actually was quite a revelation for me, that it's pustule, because there's pus in it. I think that's the advantage. Maybe if you're a native speaker, then this will be more obvious to you. And finally, liquid-filled, and again, we decide based on size. If it's a vesicle, in case it's smaller, or in case it's big, then you talk about a bulla or bulle. Yeah, so this is one way of classifying. This list is still not exhaustive, so there are still other terms that if you really go into this topic, you might want to know as well. Another way of classifying rashes could be simply by etiology or by causes, and there are three main ones. One is general hypersensitivity reactions. The important ones here are atopic dermatitis or contact dermatitis. Then the one that we'll also talk about today, infections. So we have been seen before a lot of examples for rashes caused by viral, bacterial, or also fungal infections. And the final one would be bleeding disorders. Good, let's get started now with today's topic, which was purpura fulminans caused by meningococcemia, so meaning meningococcus in the bloodstream. And also here, to get into this topic, I have one more priming question for you. In case you have studied meningococcus before, which I guess many of you have, meningococcus or Neisseria meningitidis. And the questions here are, what are its main virulence factors? And the second question is, what are symptoms of meningococcus infection? So again, you can pause the video and think about this. Okay, so what are the main virulence factors of Neisseria meningitidis? The first thing that you should think about is the capsule. Other virulence factors, pili for adhesion. Then you have also other surface proteins, which are called OPA or OPC. They are maybe not that well known or not that frequently listed as virulence factors as the pili. Then what we can also call a virulence factor is the LOS, the lipo-oligosaccharide, in contrast to other gram-negative bacteria which have LPS, lipopolysaccharide, and both of them can be, called virulence factors because of this, strong reaction of the immune system and septic shock and so on. And LOS is thought to be even stronger in triggering this immune reaction, and we'll talk about this on the next class. IgAs is another factor, so that they can remain on mucosa that secrete IgA. They can destroy this IgA. And then there are a couple more which you may or may not have heard about. So factor H binding protein, for example, which the bacteria kind of use to coat themselves with a human protein when they're in the bloodstream mainly, so they're less accessible to the immune system. Neisseria meningitidis has iron acquisition systems, which is also important for them, especially when they're in the blood. And then something which is not a factor that you can really associate with a cellular structure, but just something that you can nonetheless call virulence factor, is its antigenic variation. So Neisseria meningitidis is known for changing its surface antigens, which makes it harder for the immune system, of course, to form antibodies that would then be able to eliminate the bacterium. And in terms of symptoms, we're not going to answer this now, but this will then come clear over the next slides. The main aim of this lecture then is actually that you never again say it's just a rash. And of course there are rashes which are just rashes, but if you see something like on this picture here, which is the Purpura fulminans, then this is definitely not something where you want to think it's just a rash. And the reason for this is that the underlying condition here can be exactly what we're going to talk about today. So this bacterial infection, in particular a bacterial infection with Neisseria meningitidis, which is known to be a very fast progressing infection that can lead to death within a few hours. So if you see Purpura fulminans, you should always consider this could be caused by Meningococcemia, especially if there are no obvious reasons to believe that there's another cause. Let's talk a little bit more about Purpura, so just Purpura in general, not necessarily the Purpura fulminans, and you see two quite different pictures what Purpura can look like. Here on this left one, you could say, okay, this is maybe still somewhere in between Petechia and Purpura, or like a mixture of Petechia and Purpura, because as we said before, this is based on size. So again, Petechia is typically that you say up to 3 millimeters in diameter, and then you have the Purpura between 3 and 10 millimeters. And then if it's larger, you will talk about ecchymosis. Even so, having said that, people are not always so strict about it. So often it's also that you refer to something as a Purpura relatively independent of the size, so that people would refer to either of these as Purpura. Yeah, in general, but what you see is this redness which goes more into purple then, hence the name Purpura. And the important thing that we want to remember for Purpura is this non-blanching. So again, if you press on it, the redness does not go away. So how do you know if the redness goes away? If you put your finger on it, so you can't really see anymore if it goes away. So what doctors would do, they would take a glass slide, press on it with a glass slide, then you can look through, see if the redness goes away upon pressing. And so why is that? Why does the redness go away for certain other types of rashes? This is because there the rash essentially is just caused by increased blood flow into this area, so a widening of blood vessels, and then you can press on it and you will press the blood away, and therefore also the redness goes away. In contrast, for the Purpura, it's not just a widening of blood vessels, but it's actually a bleeding into the skin, so the blood leaves the capillaries, and not just the blood plasma, but actually the cells as well, so the erythrocytes as well. And now if you press on this, you're not going to be able to press those erythrocytes back into the capillaries, hence Purpura is non-blanching. What in general then are the big causes, the main causes for purpura? So we have on the one hand side thrombocytopenic or thrombopenic purpura, meaning not enough thrombocytes. So if there is then a bleeding in your vasculature, that means it can't be repaired, and that's the reason for the purpura. And there are a multitude of cases why somebody might be or become thrombocytopenic, starting from that those thrombocytes are not formed efficiently enough, to that they are degraded too quickly, and again, also there are a multitude of underlying causes why this could be. Second, coagulopathy, which is going to be the case then for the purpura fulminans in the context of meningococcemia, meaning that you have blood clotting, so a high level of blood clotting. Let's leave it just at that for now. And then finally, vasculitis, meaning an inflammation of the vasculature, which leads to the destruction or to damages in the vasculature. And from this point on, now we can specifically go to this purpura fulminans in the context of meningococcemia. Let's get started then with the main point for today, which is trying to explain how meningococcus in the blood, so meningococcemia, is causing this purpura fulminans, and then afterwards we'll also try to answer why is it especially Neisseria meningitidis that does this, and why are the consequences of other bacteremia often different. It starts with the LOS, with the lipo-oligosaccharide. As I said before in the priming question, we can refer to this as a virulence factor, so LOS or LPS, also often referred to as endotoxin. So it is this lipid A portion of the LOS which is recognized by the immune system and then leads to a strong immune reaction. So you may remember this. So you have chains of lipid A, which may then somehow be bound to sugar molecules. And then you will have more of these sugar molecules, which kind of forms a core oligosaccharide. And for LOS, this is then typically where this stops, so where you remain at the lipid A and the core oligosaccharides. And for LPS, so lipopolysaccharide, you would then have additional sugars outward, which then often is also referred to as this O antigen, so the thing that's recognized by antibodies and that can then be used to distinguish bacterial strains. Okay, so lipo-oligosaccharide, and people say this is stronger in terms of triggering this immune response. So immune response, mainly recognition is by monocytes, and recognition is via TLR4, so toll-like receptor 4, and these monocytes then produce these pro-inflammatory cytokines, namely TNF-alpha and IL-6. There are other ones as well that seem to play a role, so for example IL-8 and IL-1 beta. And now the typical consequence of this, the typical sepsis and septic shock would be that due to these cytokines there is vascular leakage. So this is also something that we had in other lectures already. So the junctions between the blood vessels, they get loosened, so blood can exit the vessels. Why the immune system does this is, of course, so that leukocytes, neutrophils mainly, can leave the blood vessels and go into infected tissue, but a consequence of this is also that, of course, the plasma is leaving the capillaries, but what should not, or what typically would not happen here, is that also erythrocytes leave the blood vessels in a large scale. But yeah, so the main consequence then is a loss of blood pressure, so hypotonic shock, typically because this occurs systemically, so there will be a lot of places in the body where you're losing liquid from the vasculature into tissues, and therefore, yeah, blood pressure drops. Consequence can be multi-system organ failure. So as I said, something that can happen with Neisseria meningitidis as well, but which is not what we're seeing in this Purpura fulminans. So this is more of a different mechanism, or there have to be additional mechanisms. And what seems to be going on here is that also endothelial cells react to this LOS, also via toll-like receptor 4, and they react either directly to the LOS or they react to these cytokines released by the monocytes. And what happens then is triggering of the coagulation cascade. Coagulation cascade, you may or may not know, so factors like factor 7, factor 10, thrombin, and then eventually the conversion of fibrinogen to fibrin. So these are all steps that are involved here, and also eventually the involvement of platelets. It's triggered, or seems to be triggered, mainly through this TF, through this tissue factor, but there also seem to be also other entry points, how LS triggers this coagulation cascade. And then coagulation, of course, means the formation of blood clots. Formation of blood clots leads to ischemia and finally necrosis. So how you can imagine this is you have your capillary and you would have blood flow through this capillary. Now you're starting to have a blood clot here, which completely or to a large part blocks the blood flow through this capillary. And now, of course, in all of this area, you have a lack of oxygen, so the blood can't transport oxygen here anymore, and eventually those cells will die, and these blood vessel walls get degraded, and therefore the blood, not just the plasma, but also the cells, also the erythrocytes, can leave the tissue. Okay, and this one, as I said, is the mechanism underlying the purpura or the purpura fulminans. Something else, and this is, I asked you before about typical symptoms or disease mechanisms in meningococcus infection. This can also cause DIC, so disseminated intravascular coagulation, meaning a lot of places in the body where you have these coagulation reactions. And then actually the main consequence of DIC seems to be that the body runs out of clotting factors, and then this is something that's attributed to bleeding into the skin and into tissues, that you just don't have enough clotting factors anymore. Having said that, more recent research of the last 10 years or so is arguing that for the meningococcus purpura fulminans, it may not even be necessary really to have this DIC, but that this could still occur even if there's still clotting factors present. So we'll go more into this later. This maybe is already getting a bit into the details, but definitely there's a link also between the underlying mechanisms of this purpura and the DIC, even so they don't seem to be exactly the same thing in case of meningococcus. Okay, what else can occur? Well, we know that Neisseria meningitidis is very good at crossing the blood-brain barrier, much better than other bacteria. So it's much more likely that if you have meningococcus in the blood, that this will cross the blood-brain barrier and lead to meningitis than for other bacteria. And also this seems to be associated, seems to be linked to this vessel damage, which means it's just easier for it then to exit those capillaries in the brain and get into the brain. And yeah, so typical places in the body where you have these very narrow capillaries where these kind of things happen is in the brain, then in the vessels under the skin, and also in the kidney. And finally, one more symptom associated or syndrome associated with meningococcus, more as a sequelae, so more as following the infection afterwards, is this so-called Wodehouse-Friderichsen syndrome. And here this is also linked to this massive coagulation and to destruction of blood vessels. In this case, it's the destruction of blood vessels in the adrenal gland and therefore loss of function in the adrenal gland. And the problems that are coming from this is then that you're lacking the hormones that the adrenal gland typically produce. So at this point, I think we have understood how does meningococcemia lead to this purpura fulminans. However, what we haven't explained yet is why this is specific to Neisseria meningitidis. And so if we want to go to this question, we should maybe first ask, is it really that specific? Do we see purpura fulminans only with Neisseria meningitidis? And the answer is no, actually not. We also see it with other bacteria, namely Haemophilus influenzae and Streptococcus pneumoniae seem to be important here. However, for those, it's much rarer that we see this purpura fulminans. For Neisseria meningitidis, I've read different numbers, but it seems to be at least 25% upwards. For these others, it seems to be much rarer. Nonetheless, we can ask, okay, what do these three bacteria have in common? And they have in common that they all have a capsule. What's the main role of a capsule, especially if we're thinking about these bacteria being in the bloodstream? The main role is a protection from the immune system. So particularly important here seems to be the protection from phagocytosis. So this suggests that these bacteria, well, they are especially well protected from the immune system, so therefore they can probably just reach higher numbers in the bloodstream. So we can say maybe what makes all three of them different from, let's say, other non-encapsulated bacteria in the bloodstream is, okay, they can grow to high numbers, and maybe that's why they're causing more of this sepsis symptom of the purpura fulminans. Still doesn't answer, of course, how is now the end meningitidis different from those other two, which cause this purpura fulminans much less often. In meningococcemia, the number of bacteria in the blood is high, but we also said, okay, this is something that Neisseria meningitidis has in common with those other also encapsulated bacteria. Now, Neisseria meningitidis has still a couple of more tricks up its sleeve why it can persist well in the bloodstream and hide from the immune system. But this doesn't seem the main differentiating factor for two bacteria that cause sepsis, which look symptomatically different. Also, those three bacteria, those three species that we had before, are IgAs producers. But so this also, apparently has to do more with persisting on the mucus than rather persisting in the blood in these later stages. Traditionally, an argument that's made for just higher severity from meningococcus is that the LOS, the lipo-oligosaccharide, activates the toll-like receptor 4 more strongly than LPS. However, more recently, the focus is going to something else, and this is the attachment of Neisseria meningitidis to the endothelium. And this attachment probably mainly occurs through the papillae, especially it has this type 4 papillae. And what happens then? So these Neisseria, only a few cells or only individual cells, adhere to the endothelium, but they then form microcolonies. What I find interesting is that here the diplococcal nature of Neisseria might support this colony formation, because so what's shown in this picture here is that we have this Neisseria diplococcus attached to the red one is supposed to be an endothelial cell. I'm only showing the pili here that are attached, not the other ones that are unattached. And so if we now say, okay, this cell divides, and now we say the cells split up along this vertical plane, and indeed this is the presumed mechanism for how the cell division occurs. Then we see that one of these cells still remains attached to the endothelium, and this gives the other cell time to now also attach, and then this mechanism can repeat, meaning the diplococcal nature helps with this colony formations, and the the daughter cells are not just washed away. This one I have to say is purely my own speculation, but I think it's something that makes sense, and where really this diplococcal phenotype could help with forming colonies on the inside of capillaries. So we have the formation of these microcolonies, and then this triggers signaling. So the bacteria induce signaling in the endothelial cells via beta-adrenergic receptor.
Good. So before we end,
once
as I said in the beginning,
that the main membrane underneath this bacteria.
More and more wondering if this old-fashioned type of teaching by
changes
just lecturing
in its form,
timely,
and
maybe we're
leads to structures that
every student should
protect these
have their own learning
microcolonies from the shear forces of the bloodstream.
So
So in general, it seems these microcolonies mainly form in capillaries,
so this is
bigger blood vessels, again, which is due to shear forces, and then this
Rashes,
signaling and this deformation of the cellular membrane seems to help
and
so this further resist
consists
shear
of
forces.
parts. First,
Another signaling
it's
is
supposed
to
to
say,
test
okay,
your
these are
existing
neighboring
knowledge
endothelial
on
cells, and have these
subtopic,
junctions here. Then somehow the bacteria seem to induce that these junctions
the app has
get dissolved,
your knowledge,
or
it will teach you in a Socratic
junction
manner. So
proteins
this
seem
is
to
why
end
it's
up
called
below
Socratic,
the
which
bacterial
is the
colony,
name that
and of course
Andreas
this means
gave
then
this app.
opening up
So we'll just keep going here.
of gaps
We
between the
can select
endothelial
our
cells.
topics, so different kinds
So I'm not saying this is
among
the main mechanism
rashes.
for
We
the
go to
erythrocytes
rashes.
leaving the vasculature and this bleeding into the
and
tissue, but this is something that
then you'll see this question here. So
the
6-year-old
local
child
inflammation.
presents
The coagulation
crusts,
probably
small
seems to be
vesicle
the more important mechanism here.
To space located around nose and mouth. Give the most likely
so
diagnosis.
all of this
contagious official infection
points toward that on the one hand side, this
honey-colored
attachment
crusts.
of Neisseria
So it's probably
is important in the specific phenotype of the Purpura fulminans, and also that maybe it is not so much a
now it will
systemic
process our answer,
systemic
and
damage or
we'll say that's
systemic
okay,
changes,
and
but
give
really these
more
local changes.
information about it.
So one fact that argues for this is that it has been found that strains that are defective in these type of
how well we did in the
for the
previous
attachment,
questions. In this case,
they
one previous
exhibit
question,
much
based
reduced
on this,
vascular
just
damage.
the level of these
So
questions.
this arguing for the attachment indeed is functionally
Let's maybe do
important.
one more.
If it were just
here, so
bacteria floating in the blood and
sharply
then
demarcated
there's an
red
increase
thread,
in
tended
free
mark on the right cheek.
lipid A that then causes
high fever and chills.
DIC and other kinds of cytokine storm, then we wouldn't really necessarily need this attachment. Second finding is, so this comes from a model where human
so
skin grafts were attached to mice, and then they found that there are
sharply demarcated
these bacteria, this Neisseria meningitidis, were added to the bloodstream of these mice. They found human pro-inflammatory cytokines, which argues for this local effect. From what I've seen, they didn't actually measure mouse cytokines, so it doesn't necessarily argue against a systemic effect, but at least it says, okay, there is this local effect.
What you do see here, and
I
this
would
is already one of the
extend the speculation a bit further, even so I don't really have literature for this, extend it to saying maybe in contrast to other bacteria,
of this rash,
the signals from the endothelial cells is important, and maybe not in comparison, maybe not so much the signaling from the immune cells, from the
monocytes. The second problem that of course you see is that the images are not working, which is
so let's summarize. We started talking about
with this app so far.
Rashes here, and then,
even so, I mean, of course, you have to say it's amazing that you can build an app like this with
fulminans,
programming or so on. Just
Purpura fulminans,
words telling us words,
non-blanching rash.
But nonetheless, so this for an app about rashes where it would be important to see what these rashes look like. This, of course, is a bummer.
Rash that's
originally I told AI Studio to just look at sources
quickly,
of free images and then
why?
Get
because
the from there and also include
frequently is caused by Neisseria
attribution
infection. That
this didn't work. Like these were always broken links.
Very quickly.
And then I tried to store these images locally, but it causes vascular damage through coagulation. Similarly, I told it if there are technical terms to provide a link, which it does.
In this mechanism, in comparison to
these links are also always
other
broken, so this is not
with other bacteria. It seems that this attachment of diagnosis in epithelium and those local effects are important.
The part that I do think is working relatively well is this adjusting of the levels. So if I get answers wrong, it will make it easier. If I do well, then it will make it harder. Also, if I get a lot wrong, then it will repeat similar questions, which I think is also a good thing. What I don't like so much is that it's almost always clinical cases. So I wanted to have clinical cases as one type of question, but not always clinical cases. So sometimes I would also just like that it asks for a definition or for a term or something like that. So this this is the part where it assesses the user's knowledge. So I just finished those 10 questions for assessing my knowledge on the bacterial infections. So after the last question, we click on here, final results. Then it will tell us, okay, where I did well, where I didn't do so well, in terms of subtopics of bacterial rashes. Here, kind of a score summary, and then it will suggest here, okay, you're essentially missing a lot on this subtopic, so eczema in this case, and now we will dive into an actual learning session. So let's look at this. I have to say, it took me relatively long to arrive at this point because it does think quite a lot between questions. That's probably because it tries to assess my knowledge and then adjust the level of the questions. But this is another reason, where I would say this is not so great yet. It takes a bit long. Okay, let's see here. Okay, so it says, okay, let's think about eczema. I hope you have read it yourself, but essentially think about eczema in terms of how deep the infection is, and it contrasts it to impetigo. And so now eczema is considered a deep form of impetigo. Which layer of the skin do I think that the infection must penetrate? Okay, so the bold one doesn't really work here, but I can ask about this for a definition. So I would say must penetrate the dermis would be my guess. So it asks how does it regenerate differently than something where only the epidermis is affected. I guess would be maybe there is fibrosis. Okay, so this seems to be okay as well. Then it continues to the who, so which bacteria. Yeah, I have to say I haven't played with the second part too much yet with the well, what it calls Professor Feynman or Socratic teaching technique. Overall, so far my impression of this is pretty good. Well, maybe a bit like other chatbots as well, which are quite good at teaching. So I think it's doing that part pretty well. I also had times where, I didn't really find the answer and where it then led me towards it. And so I think, yeah, this part is pretty good. So as you see, overall, I mean, it still is amazing that you can build these kind of tools nowadays without programming. I definitely think the future of learning is going in that direction. So rather than sitting in classes, to have your personal AI tutor. However, still, I mean, this tool isn't quite there yet. Main thing, probably the pictures, which are not displaying. But yeah, so I just wanted to give you this little attempt towards personalized teaching.
Yeah, so I haven't tried to publish this app yet or anything, or to make it available because, as you see, there are quite a few issues still with it, which I think makes it not really useful yet. But yeah, I would be definitely very interested to hear your comments. Maybe you have suggestions how this can be done better, or maybe using a different tool than Google AI Studio or something like that. Okay, so finally these two are the main references that I used here. So both of them are reviews about 10 years old on the pathogenesis of meningococcemia. And last but not least, you have seen there were several images, mainly of rashes here. So these are the image credits. Almost all of them are coming from Wikimedia Commons. And so, yeah, huge thanks as always for people who provide these images. I think that's, yeah, hugely important, and would definitely be better if we still had more freely available medical images. Thank you, and till next time.